In this next session, we're going to be looking at bioenergetics and metabolism. So by the end of the session, you should be able to understand how energy is transformed through the biochemical reactions and what sort of energy forms can be kind of taken on by the body. We're also going to look at thermodynamic differences between exergonic and endogonic reactions, so you should be able to define these. And also to define metabolism and what is metabolism, and describe how the metabolic pathways are interrelated and regulated. First, we're going to touch on dynamic state, steady state. So living organisms must perform work to kind of stay alive and to be able to reproduce themselves. So they need to have a system in which they can take food and break that food particles down and to make energy. There's a kind of characteristic composition of the organism which kind of changes through time, and that's through kind of metabolic regulation, but the population of molecules within it is far from being static. So there's small molecules. So we've kind of visited macromolecules. We've looked at things like carbohydrates. We've looked at proteins, lipids. We've also got supramolecular complexes, which are kind of continuously being synthesized and broken down in kind of chemical reactions that involve constant flux of mass and energy. This is known as dynamic steady state, and it's far from the equilibrium within its surroundings. So maintaining the steady state requires a kind of a constant investment of energy, okay? And when a cell can no longer obtain energy, this is the point when it decides it has to die. And so it begins to then decay and towards an equilibrium within its surroundings. Have a look at this video later on. This kind of explains the difference between equilibrium versus steady state. So the biological energy transformations kind of obey the laws of thermodynamics. And what the first law of thermodynamics says is that conservation of energy, so for any physical or chemical change, the total amount of energy in the universe remains constant, and energy cannot be created or destroyed. So, for example, motion. Motion is a mechanical energy. Energy can be taken from light, so light can also be energy. And also heat. Our bodies are heated to 37 degrees. This also requires energy. And also we have chemical energy. So cells are actually kind of consummate the transducers of energy, and they're capable of interconverting the chemical, the electrical, the osmotic and mechanical energy with great efficiency. If we look at the second law of thermodynamics, so creating, maintaining the order requires the work and energy. So the second law of thermodynamics tend towards increasing disorder. So in all natural processes, entropy, denoted as S, of the universe increases. If we look at our macromolecules, so things like DNA and RNA, proteins, these provide information. So there are informational macromolecules. And there's actually a precise organization of these subunits which then contains this information. In addition, using energy, we can make these molecules, and they can invest energy to make sure that the subunits are in their kind of correct sequence. So, the molecules are highly ordered, but they're actually violating the second law of thermodynamics. Bioenergetics. Bioenergetics analyzes energy changes, and so we need to focus on the well-defined part of our universe. Essentially is the system. So the system could actually be the cell. okay, in the simplest form. If we then consider the environment, so the surroundings, so this is outside the system, okay, the surroundings. So say if we take, for example, we have different types of systems, so we have the isolated, and there's no energy or mass exchange. In a closed system, there is an energy exchange, but there's no mass exchange. Whilst in an open system, there is energy and mass exchange. This we constitute as a living organism. And living organisms, they are never at equilibrium with the surroundings. So there's always a constant change between the system and the surroundings. And that explains how organisms can create order within themselves while kind of operating within the second law of thermodynamics. So free energy. So free energy essentially is the content of that closed system, which can be defined in the terms of free quantities. First, we have randomness, known as entropy. And then we have the number and kind of bonds, which is known as entropy. And then we also have absolute temperature in Kelvin. This is known as Gibbs free energy. So Gibbs free energy equals entropy minus absolute temperature in Kelvin times entropy. So when a chemical reaction occurs at a constant temperature, the free energy change is kind of determined by the entropy change, which is known as delta H. And that kind of is reflected as numbers and kind of kinds of bonds, so chemical interactions, which can be created or formed. And the change in entropy is actually describing the system's randomness. So if we look at this equation again, kind of rearrange it, delta G is the free energy equals delta H, which is entropy change, minus the temperature in Kelvin, and then times delta S, which is the entropy. So by definition, delta H is actually the negative for a reaction that kind of releases heat, whilst delta S is actually positive for the reaction that kind of increases the system's randomness. First, the spontaneous reaction of delta G is a negative free energy, which is then released. So what the cell does, it functions by using these molecules, and there is like free energy formation. The free energy formation is positive. So the molecules that are formed are actually less stable, and they're actually more highly ordered than their monomers. So how do they form? If we relate the free energy to macromolecule synthesis, we can carry out energetically unfavourable energy, which requires endergonic reactions. And the cells kind of couple them to reactions that liberate free energy, exergonic. So the overall process is exergonic. So essentially the sum of the free energy changes is negative. The source of energy, the usual source of energy, is a kind of is normally coupled to a biological reaction, and what happens is the energy is released by breaking something called the phosphoanhydrate bonds, which we find in adenosine triphosphate or guanosine triphosphate, so ATP or GTP. ATP is a very common source of free energy. We also have energy coupling reactions. So bioenergetics is kind of concerned with the study of energy transformation in the living system. So what this means is that the free energy from the fuel metabolism or light is captured in a kind of coupled system in the cell's energy requiring function. So you can see in the graph here we have free energy, which is denoted as G, and then the reaction coordinates. So in reaction 1, here we have glucose plus phosphate, which makes glucose 6-phosphate. Here this requires, so this is the addition of a phosphate, this requires energy, which is denoted as delta G1. In reaction 2, this is the breakdown of energy, so our energy molecule here, which is ATP, to ADP plus phosphate. So this requires then essentially a depletion of the energy, which is denoted as a delta G2. And in reaction 3, we have glucose plus ATP. So here, glucose plus the ATP gives us then glucose 6-phosphate plus ADP. Now the difference in the energy is G3 equals delta G1 plus delta G2. So it's the difference between these two reactions to give us the overall energy state. If we consider a reaction in a closed system, the chemical reactions proceed spontaneously. Essentially, they will proceed until the equilibrium is reached. Here we have the reactant, which then will proceed to make the product, and there is an equilibrium. Okay, so the equilibrium will proceed until the chemical reaction, when the components in the reactants run out or the products are broken down. Now at the equilibrium, the forward rate equals the reverse rate. So R1 equals R2. At equilibrium, the rate at which the reactant is turned into a product is the same rate at which the product is then turned back into its reactant. So in this case, there is no net change in the concentration of the product or the reactant. And the energy change as the system moves from an initial state to equilibrium with no changes in temperature or pressure is given by the free energy, so delta G. And the magnitude of delta G depends on the chemical reaction which is involved and how far the equilibrium system is initiated. Next, if we take the kind of concentrations of reactants, so how much we're putting into the reaction, and the products at the equilibrium. This is defined by the equilibrium constant, known as K equilibrium, or the equilibrium constant. Here in this equation, we have small a, which is the number, so times large A, plus small b times b, gives us then small c times c plus small d times d. So in this case, A, B, C and D are the number of molecules, whilst the capital letters are A, capital B, capital C, capital D. These actually participate in the equilibrium constant. Now we can actually rearrange this in as part of the equilibrium constant. So what this means is, here we put the products on the top, and in the bottom we put the reactants. So here we have large C times, so the concentration of C, times D, concentration of D, over A times A, B times B. So in this case, in the brackets, this is the molar concentration of our products over our reactants. And it's the molar concentration of the reactant components at a point of equilibrium. So when a reacting system is not in equilibrium, the tendency of the reaction is to move the equilibrium in a kind of driving force. And the magnitude, if we can then be expressed as free energy change for the reaction. If we consider the standard free energy change, the standard free energy change is defined under the following conditions. It's defined by temperature at 25 degrees, which gives us, we work in Kelvin, so it gives us 298.15 Kelvin. Partial pressure of gases, which is 1 atom, or 103.3 kilopascals. And the reactant and product concentrations initially is 1 molar. So under these conditions involving hydrogen ions, the reactions are actually involving hydrogen ions, which would be 1 molar or pH 0. So the biochemical reactions that occur in a well-buffered aqueous solution near pH 7 with a concentration of water would be 55.5 molar and magnesium concentration at 1 millimolar. This essentially is the biochem's standard state. In a standard transform constant, the temperature is 25 degrees, okay? The partial of gases 1 atom, reactants and product concentrations at 1 molar, pH 7. Water is at 55.5 molar and magnesium at 1 millimolar. So the constants, all at the standard biochemical conditions, incorporate the concentrations of water, hydrogen and magnesium. If we consider the free energy, this table shows you the relationship between standard free energy changes of a reaction and equilibrium constants. At an equilibrium constant of 1, the free energy change is 0. Whilst if we look at the equilibrium constant at 10 to the power of 3, we see that the free energy is in a minus range. If we look at the equilibrium constant at 10 to the minus 4, here the standard free energy is in a positive range. So there's a relationship between the delta G and the equilibrium constant. So the standard free energy change of a chemical reaction is simply an alternative way of expressing its equilibrium constant. We can express it as this equation. Here we have delta G equals minus R, which is the gas constant, which is always 8.314 joules per mole, IN, and then K is the equilibrium constant, and T here is the temperature in Kelvin. When the equilibrium constant is less than 1, then the delta G is in the negative range. So the reaction will proceed spontaneously forward. So it will move forward through that equilibrium. and the products have less free energy than the reactants. When the equilibrium constant is less than 1, then the delta G is in the positive range, and the reaction will proceed spontaneously in the reverse, and the products have a greater free energy than the reactants. And when the equilibrium constant is equal to 1, then the delta G is equal to 0. And there is a reaction which is then at equilibrium. And the products and the reactants have equal free energy. And the actual free energy changes. So it changes because it's dependent on the reactant, the product concentrations. It could also change because of temperature. But we have to be careful to distinguish between actual free energy changes of delta G and the standard free energy changes. If we take an example of a calculation, so here we have glucose 6,1-phosphate is an equilibrium with glucose 6-phosphate. The initial concentration we have in the reactants is glucose 1,6-phosphate, is 0.02 molar, and glucose 6-phosphate is 0 molar. The equilibrium concentrations that we observe are glucose 1,phosphate is 0.001 molar, whilst glucose 6-phosphate is 0.019 molar. So does the reaction proceed to glucose 6-phosphate with a loss or a gain of free energy? What do you reckon? Well, in fact, if we calculate this, what we observe is that the equilibrium constant of glucose 6-phosphate over glucose 1-phosphate, remember the products and then the reaction, we can then put these values into the equation. So remember that glucose 6-phosphate was 0.019, we put this here, which is the products, and the glucose 1-phosphate was 0.001 molar, which gives us 19. So as the reaction is at equilibrium, we can use the standard free energy equation. And if we use the standard free energy equation, which is equals minus RT in equilibrium constant, gives us then the gas constant, minus 8.314, times temperature in Kelvin, times 298.15, times in and 19, which gives us then the free energy, which is minus 7,298 joules. So this reaction actually proceeds spontaneously with a release or loss of free energy. So in fact, this reaction is there is a loss of free energy. So the actual free energy change for a reaction essentially is a function of a standard free energy change. It's dependent on the reactant and the product concentrations, and also of the temperature of the reaction, and none of which may be standard at standard conditions. So for our reaction, we have small a times A plus small b times b gives us then small c times c plus small d times d. So delta G and the standard free energy change can be related by the following equation. So here we have the free energy equals the standard free energy change plus RT in, and remember we put the products here and the reactants here, and these are like concentration times C, concentration of D times D, etc. Here, this is the actual free energy, as I said, the standard free energy change. Here's the gas constant, 8.314, and that's the temperature in Kelvin. And this essentially is called the mass action ratio, and it's simply the ratio of product concentration to the reactant concentrations. And we simply enter the actual concentration of A, B, C and D. So these are actually the actual reaction conditions. Okay, so we have the gas constant times the temperature of the reaction, and then we have here are the products and then the reactants. So the free energy change is a measure of the distance of a system, so from its equilibrium position. So when at equilibrium, the free energy is equal 0. At equilibrium, there's no driving force on the reaction in either direction. So delta G equals 0. Here we can then simplify this equation. We essentially then, where there's no equilibrium change, we essentially remove this part and then simplify this equation. So this is then the actual standard biochemical conditions. In example 1, we're looking at energetic cost of ATP synthesis. So if the equilibrium constant for the reaction ATP, the hydrolysis to ADP plus phosphate, is 2.22 times 10 to the 5 molar. Next, now calculate the standard free energy change for the synthesis of ATP from ADP and phosphate at 25 degrees. So now remember, the standard free energy change equals the universal gas constant. Remember, the gas constant is 8.315 joules per mole times the temperature in Kelvin. So we need to convert the temperature of 25 degrees into Kelvin. And at 0 degrees, there is 273 Kelvin. Okay. So the solution is to first calculate the free energy change using the equation above. So free energy change equals minus, in brackets, 8.315, which is the gas constant, times the temperature in Kelvin, so 25 degrees, is 298 Kelvin. And then times in, which is then the concentration, okay, 2.22 times 10 to the 5, okay, equals 30.5 kilojoules per mole. And remember, we asked to calculate the free energy change, the synthesis of ATP, so then the reaction is reversed. So from ADP plus phosphate to ATP. So if we reverse the reaction, we change the sign, okay, of the standard free energy value. So it goes from minus 30.5 kilojoules per mole to 30.5 kilojoules, so as a positive figure. In our next example, here, example 2, we're looking at glycolysis, so the conversion or the interconversion of fructose 6-phosphate to glucose 6-phosphate. Has an equilibrium constant of 1.97. What is the standard free energy change for the reaction, assuming the temperature is at 25 degrees? And if the concentration of fructose 6-phosphate is adjusted to 1.5 molar, and glucose 6-phosphate adjusted to 0.5 molar. What is the free energy? So the solution, so at equilibrium and standard conditions, therefore you can use the standard free energy change equation, which is this, remember. So here we have the gas constant, remember R is the gas constant, times the temperature, so 25 degrees, 298.15 Kelvin, times in, and then times the equilibrium constant, which was 1.97. That gives us then minus 1,680 joules. As the reaction is no longer equilibrium, you need to then use free energy equation to determine the mass ratio. So the mass ratio is 0.5 divided by 1.5, which gives us then 0.333. So the free energy change is equal to the standard free energy change plus then the gas constant times temperature, in and Q, which then equals minus 1,680 plus 8.314, which is the gas constant, times the temperature in Kelvin, 298.15, times in, which is the mass ratio, 0.33. This gives us then a result of minus 4,403.3 joules. So now moving on to pathways. So pathways are used in the cells. They're used for kind of biological processes. They help to regulate different mechanisms, and they are used by thousands of enzyme. So essentially, there are thousands of enzyme catalyzed reactions that are going on in the cells. Now, these have to be orchestrated in the correct way, and so they have to be organized into kind of sequence events, so consecutive reactions, which then are called pathways. So if we take, for example, E. coli synthesize amino acids, leucine from threonine. And this is done in 5 steps. Here, for example, we have threonine, enzyme 1 reacts with A to produce B. B reacts with enzyme 2, C reacts with enzyme 3, D reacts with enzyme 4, E reacts with enzyme 5, then to produce isoleucine. So the metabolic regulation, so when we produce this product, once we produce this product, there is a feedback effect, okay? The feedback effect will act on various parts of the pathway. So it may act on the beginning of this pathway, so it will act on a particular enzyme, so enzyme 1. So not only do cells actually simultaneously synthesize thousands of different kinds of metabolites, but they also do this in a precise measure. So they make them according to requirement. So if a cell kind of begins to produce more isoleucine than it needs, then the unused isoleucine then accumulates in the system, and then the increased concentration then inhibits the first enzyme on the pathway. And that's immediately what happens then, it immediately then slows down the production of isoleucine. So there's a feedback, we call this a feedback effect. And this feedback effect is used in our cells in all these different pathways. And these feedback effects help to manage metabolic metabolism. If we consider how metabolic regulation occurs, isozymes are different types of enzymes that actually catalyze the same reaction. So if we take aspartate can be actually catalyzed by 3 different types of isoenzymes, which we denote here as A1, A2, and A3. The same is also true with aspartate beta semialdehyde. That can also be catalyzed by 2 different enzymes, which produces homoserine. Threonine can also be catalyzed by 2 isozymes, C1 and C3. What you notice is there are feedback effects. So when we accumulate a particular product, there is a feedback effect on a particular isozyme. And this actually allows for kind of interlocking regulatory mechanisms to kind of fine-tune the amount of products. Isozymes may actually have different affinities and also the rates of reactions, or are inhibited by different molecules. This then ensures that there is a supply of important intermediates which are used by other pathways, which is not completely depleted. So it's actually trying to be kind of cost effective in energy requirements to produce enough products, but not to produce too much. So the flux through an enzyme catalyzed pathway can actually be modulated by internal and also external signals. This modulation allows modulating catalytic activity through different types of interactions. This might be through allosteric changes in the enzyme catalysis. So these are internal signals. So things like substrate concentration can alter the internal signal, cofactor concentration. Also key metabolite concentration. It can also be caused by external signals, so the concentration of a cellular second messenger, so things like cyclic AMP or calcium divalent ions. For changing number of enzyme molecules, enzyme synthesis may be involved. So this will change transcription rate, transcription factor activation, in the case of the nucleus turning on different genes, translation rate, so the production of proteins, and also the stability of messenger RNA. So messenger RNA can be degraded. An enzyme degradation rate and turnover can also change. And there's also a compartmentation of specific enzymes away from the substrate. So reactions far from the equilibrium are at kind of a common point of regulation, i.e. the mass action ratio, Q, which is very different, and it's greater than 2 orders of magnitude from the equilibrium constant. So there are usually 2 reactions involved in a kind of fundamental cellular process, and if they're allowed to be to complete the equilibrium would then be disastrous for the cell. So cellular metabolism is governed by 2 main pathways. We have anabolism, which is a pathway that kind of starts with small precursors, so such as amino acids, sugars, fatty acids, nitrous bases. These then produce those like kind of large macromolecules that we know about, proteins, polysaccharides, lipids, nucleic acids. The production of these macromolecules requires energy, okay? And the energy is actually made through catabolism. And catabolism essentially is taking those food molecules, the carbohydrates, the fats, the proteins, breaking them down to essentially waste products. These are kind of energy depleted end products, so things like lactic acid, carbon dioxide, water, ammonia. And there's like a degradation of these organic molecules, which then releases energy. And that energy can be released as ATP, NADH, NADPH, and FAD-H2. These are our chemical energy stores. And these chemical energy stores then are used to produce our macromolecules. And so the chemical energy released is used to synthesize ATP, and also cellular ATP is then held far above its equilibrium concentration, and the free energy for ATP breakdown is large and negative. If we consider anabolism, these pathways require the use of energy, and so they actually consume ATP and other energy molecules, so NADH, for example. So overall, the network of the catabolic and anabolic enzyme catalyzed pathway constitutes cellular metabolism. And the organelle responsible for the kind of catabolic pathway is the mitochondrion. So the mitochondrion in the higher organisms essentially converts the energy stored in those food molecules into free energy in molecules of ATP. And we have ATP synthase here. ATP, also known as adenosine triphosphate, is a really important compound because it transfers phosphate groups. So ATP contains these two phosphoric anhydride bonds and one phosphoric ester bond. So we have the triphosphate here, which is then linked to the ribose and then to the base, which is the adenine. ATP undergoes hydrolysis of the terminal phosphates. So you have hydrolysis of the terminal phosphate, which then gives us ADP. Okay, so it's adenosine diphosphate, still has two phosphates. And energy. The hydrolysis of the phosphoric anhydride liberates more energy than the hydrolysis of the phosphoric ester. And so we say that ATP and ADP contain two high energy phosphoric anhydride bonds. ATP is then the universal carrier of phosphate groups and is also a common currency used for storage of energy and also transfer of energy. And the energy generated from this reaction, which would be about 7.3 kilocalories per mole. NAD, also known as nicotinamide adenine dinucleotide, is also a biological oxidizing agent. If we look at the structure of NAD, the plus sign here denotes on NAD plus is the positive charge. It's the positive charge on this nitrogen in the part of the nicotinamide. There is also an N-beta glycosidic bond here, and also we have the nicotinamide, which is then derived from niacin. NAD plus is a two electron oxidizing agent. It is reduced to NADH. NADH is a 2 electron reducing agent, and this can then be oxidized to NADH, so NAD+. So NADH is an electron and hydrogen ion transporting molecule. FAD, or FADH2, also known as flavin-adenine dinucleotide, is also a biological oxidizing agent. So the structure of this includes a flavin part. We also have a ribitol part, and this part here constitutes as the riboflavin part. FAD is also a 2 electron oxidizing agent and is reduced from FAD to FADH2, and the FADH2 is a 2-electron reducing agent, and this can be oxidized to FAD. We need to know about these energy molecules because they play an important role in those metabolic pathways. And we're going to be discussing these metabolic pathways in the coming sessions. So the metabolic relationships within the cell essentially allow energy to be made, but also the metabolites to be made in the cells. And there are different pathways. There's a kind of convergence or linking of those different pathways, which then gives those key intermediate metabolites. So we have converging catalysis, which feeds into the citric acid or Krebs cycle. And it also feeds into a diverging anabolism. The diverging anabolism produces metabolites such as the hormones, the bile acids, cholesterol, vitamin K, the triglycerols. The starting material here is the foods, so things like starch, glycogen, sucrose. They're kind of broken down into kind of smaller products like glucose, which then feeds into the pyruvate, which then feeds into acetate, acetyl-CoA, which then goes into the citric acid cycle. So there is a linking of all these pathways, and those pathways, if we were to look at them globally, it's very complex, but they all have, essentially they're producing different metabolites which can be used in those different pathways. So the science behind understanding how each of these metabolic pathways interact with each other is quantitatively called systems biology. And metabolics is essentially the global study of those metabolic chemical intermediates within the cell at a precise moment. And these large-scale experiments help to unlock how the cell is controlled and which pathways are important in disease states, for example. Now we can have a look at the interactive metabolic pathways, and we can use some databases. And there are different internet based interactive pathway maps which help us then to interrogate those particular metabolic pathways. And a common one is the KEGG pathway. We can have a look at this in the session. But these pathways, as you can see just from this picture, this shows you all of those metabolic pathways. And it's very complex, but we can look at those different pathways and see how those pathways are interacting. Anyway, that comes towards the end of the session. I've identified some kind of further reading for you. So there's a book chapter in Lenin Principles of Biochemistry, 6th edition. You can have a look at part 2, so Bioenergetics and Metabolism, pages 501 to 504. Also, if you look at pages 505 to 528. In Berg et al., Biochemistry, chapter 15, Metabolism and the Basic Concepts and Design, have a look at pages 223 to 444. Anyway, thank you for listening, and I'll see you in the session.